Hey guys, it's Professor Tiffany Martin with Medical Microbiology, and we are working through Chapter 16 and the adaptive immune response. So we kind of did an introduction, and I kind of described it as these are the big guns. These are the ones that are really targeting a pathogen and attacking them. So how do they target the pathogen? How do they recognize the pathogen? We need to talk about antigens. So let's do that. All right, what are antigens? Antigens are molecules, and these are going to be on the pathogen that our body recognizes as foreign and worthy of attack. So let's go ahead to the picture and then we'll come back to this. So we are being attacked. We have passed our physical barriers. We have something inside of us. Our second line of defense is going to do some things to immediately to try to protect us, but we need to determine our adaptive immune response, our T cells and our B cells need to determine if we need to specifically attack this pathogen. Is it bad? So we need to recognize it. All right, so here's my pathogen. It's unfortunate, it's not a bacteria because it has a nucleus, but maybe it's a microorganism, maybe it's a fungus, maybe it's a yeast. Either way, it got in our body and we need to see if it is bad. All right, so here's the cell. If we're going to blow that up on the cell, you've got your cell membrane there for both prokaryotes and eukaryotes, the cell membrane, and it's going to have those integral proteins. There's going to be something on the surface of this pathogen that the T cell or B cell will recognize. This is what we call an antigen. Now it's got a proper name. It does something for this microorganism. You remember when we talked about how when we did our representative pathway for our metabolic pathway, and we talked about precursor and end products and intermediates and byproducts, and we said that it had a general name. Now this antigen is its general name when it's talking about in its relationship to the immune system. So we're going to call this particular protein on this pathogen an antigen because it's what the immune system is going to recognize and bind to. Now, just like our enzymes in an active site, it's not going to bind the entire protein. Part of the protein is inside the cell. It's actually going to bind a specific spot on that protein. So it's an even smaller place. This is a protein with a whole bunch of amino acids. This may only be like 3 or 4 amino acids. It's a very small section, but that is the section that's going to actually come in contact with our adaptive immune system, this T cells or the B cells that are binding to this. Those are referred to as epitopes. Another name for epitopes is antigenic determinants. Okay, so understanding what an antigen is, we'll go back. What it is and what it's located, it's just a protein that this microorganism makes that happens to be on the outside of the cell, but we refer to it as an antigen because it's where the T cell or B cell is going to bind. The specific place where it's binding is called the epitope or the antigenic determinant. All right, back to our slide here. Antigens are molecules, proteins, lipids don't make really good antigens, proteins, sugars, something that's on the cell surface that our immune system is going to recognize as foreign, worthy of attack. It's recognized by these three-dimensional regions called epitope. So that's the little piece of the antigen that it recognizes. Another word for epitope is antigenic determinants. Things that are foreign, macromolecules, things we don't have make the best antigens. Because remember, we take in things all the time. Food, dust. We don't want our body pulling out the big guns and shooting up our food. That's not what we want. So there's lots of things that our body takes in that's foreign that does not deserve an immune response. And so your body has to learn to tell the difference. So it does things that are particular to pathogens make good antigens. Pieces of viruses, fungus, protozoan, those are different than us, bacteria, those make good antigens. Unfortunately, some people's bodies do respond to food or dust with an immune response. And these are allergies. This is why some people can be allergic to shellfish. Their body has decided when they bind to that antigen that it's worthy of an immune response, and it will do an allergic reaction, which could be, something from a rash or hives or shortness of breath to anaphylactic shock. So an over response of the immune system is not good. Again, these are the big guys. These are the ones we want to be very careful before we turn them on ourselves. All right, so those are antigens. What else do I need to tell you from antigens? I kind of described a pathogen. This one could be a bacteria, it's a prokaryotic cell. Pathogen gets into our blood past our first line of defense. Our second line of defense is our responding, but we need to find out if it's worthy of our third line of defense. So the antigens are located on the pathogen, just like we showed in that last picture. There's my antigens with their epitopes. This is a piece of a flagellum. We don't have a prokaryotic flagellum. These make good antigens that our body will bind to. This is a virus. The glycoproteins on the virus may make good antigens. We actually classify this a little bit more as exogenous antigens because our immune cells are going to find it on the outside of the pathogen. So we refer to these as exogenous antigens. But what happens if this pathogen or this virus, this pathogen, has already gotten inside one of our cells? So this right here is my body cells with its own proteins and its own job, but it's now infected by a virus. Our T cells are designed to find these guys. How does it know? Once this virus has gotten inside, my T cells running through my lymph system, my circulatory system, how are they going to find these guys? Well, this cell needs to let us know, let the immune system know that it's got a virus inside. So it does that by taking pieces of these viruses and putting it on the outside of its cell. We refer to these as endogenous antigens because it came from inside the cell. Now, again, it's the same thing, maybe the exact same thing, but where is it located? This virus has already gotten inside of a cell. The cell is going to take a piece of that virus and put it on the outside of its cell membrane, kind of as a flag, going, hey, come look here, I'm invaded, come take care of this. It's a flag to our T cells and our adaptive immune response. All right, so these are endogenous antigens. They came from inside our cells. They're still the virus. They came from inside our cells, got processed, and are now on the outside of our own cell. Endogenous antigens. Now, the last type of antigen, we refer to as autoantigens. Remember, we do not want our T cells and B cells attacking our own cells, but sometimes it happens. Diseases like lupus, rheumatoid arthritis, Crohn's disease seems to have an immune component to it. This is when our own body starts attacking its own cells. So this is our own body cell just doing its own business, having its own proteins that do a function for the cell, but for some reason our immune system has decided this is bad. It will bind to one of these proteins on the outside of our normal cell and start to attack it. And again, that's when you get those autoimmune diseases that are so very hard to treat. We refer to these as autoantigens. Again, just a normal protein that you normally make that's doing its normal job, but because of its relationship to the immune system, because a T cell or a B cell can bind to this, it's referred to now as an autoantigen. Okay, all right, so we will stop that video there. Review antigens, make sure you understand that, and let me know if you have any questions, and then we'll pick up with the next one. Thanks, guys.